So now let's take a look at the basics of neural activity. So don't forget, neural activity is just a neuron communicating with another cell. And there's electrical events and chemical events. So there's going to be three main electrical events that we need to occur during neural activity that lead to one chemical event, right? Resting potential, graded potentials, and action potentials are all electrical events that lead to synaptic activity, which will be our chemical event. So here we see a neuron at resting potential. So this would be step one. We have to start off at rest in order to do the other events. So we're at negative 70 millivolts and we're not really doing anything. We're just being a little neuron, just kind of chilling, okay? The next thing that would happen is step two, the graded potential. A graded potential is caused by a neurotransmitter binding to the neuron, causing a change in voltage in that neuron. Okay? So you can see the voltage changed from negative 70 to negative 65 millivolts. Step three is our action potential. With an action potential, we get a very big change in voltage. Notice we went up to plus 35 millivolts. And this is a big change in voltage that travels down the axon. Okay? And when the action potential gets to the end of the axon, that will lead to step four, synaptic activity. So step four is our chemical event, where we release neurotransmitter. The neurotransmitter will go and bind to another neuron, causing a graded potential, action potential, and synaptic activity to possibly occur in that neuron. So again, resting potential is where we have to start off at. Neuron has to be at rest in order for the next steps to occur. A graded potential involves a neurotransmitter binding to its receptor, causing a small localized change in voltage in the transmembrane potential. Graded potentials may or may not lead to an action potential. We talk more about that in our graded potential videos. Assuming it leads to an action potential, we would head into step three. Don't forget, an action potential is just an electrical impulse traveling down an axon. It's also known as a nerve impulse. Action potentials are started by certain types of graded potentials. Action potentials are very dramatic, big changes in voltage, unlike a graded potential, and they're moving. Graded potentials stay localized, whereas action potentials travel all the way down an axon. Once the action potential reaches the end of the axon, it causes the release of neurotransmitter during the synaptic activity portion. So during synaptic activity, again, the neurotransmitter is going to be released due to the action potential. The neurotransmitter will bind to another neuron and then cause a graded potential in that neuron. So now let's take a look at resting potential. Resting potential is also known as resting membrane potential. So this is our baseline. This is where the neuron starts off at in order to experience the graded potential, action potential, and synaptic activity. Ion distribution is really what determines resting membrane potential. So ion distribution is also what will determine the direction of ion diffusion if we open a channel, which will be necessary for graded potentials and action potentials. So in order to understand all of this, we really need to know which ions are located where, okay? So the main ion players in neural activity include sodium ions, calcium ions, chloride ions, potassium ions, and then proteins, which are a large negative ion. So this picture kind of sums up what you need to know, okay? On the outside of the neuron is where we typically find sodium, calcium, and chloride. On the inside of the neuron is where we typically find potassium and proteins. I say typically because that doesn't mean that all of the potassium is inside of the neuron. There will be some potassium outside, and not all of the calcium is going to be on the outside of the neuron. There's some calcium inside. But for the most part, this is the distribution. I have a little memory tool in order to help remember this listed here. A student from a previous semester came up with this, so I can't take credit for it. So it's 1 nervous calico cat is kangaroo phobic. So the 1 for outside, N for nervous for sodium Na, calico, calcium, cat, chloride. Is, the I in is for inside, K in kangaroo for potassium, and then the P in phobic for protein. So resting potential is about negative 70 millivolts for neurons. Again, don't forget it's negative because the inside of the neuron is less positive, aka more negative than the outside. It's 70 because the actual voltage difference between the inside and the outside is 70 millivolts. So there are a couple of things that play a major role in influencing this resting membrane potential. Proteins are negative and they're inside of neurons, and that plays a major role. And then there's the sodium-potassium exchange pumps in the cell membrane, which are really important too. There's a third component that's important for resting membrane potential. These are ion leak channels, but I don't believe that they are essential for understanding neural activity at our basic level, so I'm not going to talk about them. But let's talk about the proteins and the sodium-potassium exchange pumps. So first up are the proteins. So the proteins are a major player in resting membrane potential. Proteins are negative ions or anions, and they're almost exclusively located inside of neurons. About 94% of the proteins are inside of our cells, inside of our neurons, and not outside. So this is huge because this is contributing massively to that negativeness on the inside of the neuron, right? Recall that the resting membrane potential is negative 70 millivolts. We always talk about what the inside looks like compared to the out, and these proteins are negative, making this a negative resting membrane potential. The second component is a little bit more complicated. So these sodium-potassium exchange pumps are really important for resting membrane potential. They're really important for getting back to resting membrane potential once you've finished things like graded potentials, action potentials, and synaptic activity. So sodium-potassium exchange pumps are active transporter proteins embedded in the cell membrane of all of your cells, but they're really important for neurons. So you have hundreds of these sodium-potassium exchange pumps embedded in every neuron. And they're doing what it sounds like they're doing. They're exchanging sodium for potassium, right? If you recall active transport, pumps are a typical active transporter. They are moving things against their concentration gradient. So we're moving both sodium and potassium here against their concentration gradients. And we're not moving sodium and potassium at a 1-to-1 ratio. We're moving it at a 3-to-2 ratio. We're going to pump 3 sodium ions outside of the neuron for every 2 potassium ions that we pump into the neuron. And this differential in the ratio is really important because we end up putting more positive ions outside. 3 positive sodiums go out for every 2 positive potassiums that come in. So we end up with more positiveness on the outside. And again, resting membrane potential is negative on the inside compared to the out. So the fact that we're putting more positive ions on the outside helps with this negative resting membrane potential on the inside. These sodium-potassium exchange pumps are super important for neural activity, and I cannot emphasize this enough. Scientists estimate that 50% of your food energy, 50% of all of the calories that you eat, go to making ATP to run these active transport pumps. That's how important they are, right? So when you run out of food energy and your brain gets foggy, you know why, right? You're not using these sodium-potassium exchange pumps properly. Now, we need to put all of that sodium outside and potassium inside so that we have a gradient for sodium to come into the cell through diffusion and a gradient for potassium to leave the cell via diffusion. And this will become more important when we talk about things like graded potentials and action potentials. So that was a bunch of words. Here's a picture that might help. So again, we can see these key players in resting membrane potential, right? Including which ions are located where. And then now I've added in our sodium-potassium exchange pump. So don't forget the sodium-potassium exchange pump requires ATP energy, and it's responsible for putting all that sodium outside and for putting all that potassium inside. So here's a couple questions. I want you to check your learning, pause the video, answer the questions, and then when you're ready, go ahead and continue playing the video to see if you got your answers right.